Hello, everyone. It's a pleasure to be here. We are going to present our research called Disrupting Herpes Virus Investigation in Lunar Orbit, a System for Animal Cells Analysis. Also, here we have the names of the members, and well, we start the scientific explanation of the project.
About the mission's impact and its importance, Epstein-Barr virus is a member of the herpes virus family, which infects more than 90% of world's human population. This virus coexists within most human hosts without causing serious consequences, but in some individuals, this virus is implicated in the development of malignancy, since it has been directly related to carcinogenesis. According to this, the International Agency for Research on Cancer classified it as a type 1 carcinogen, because infected cells would have the potential to develop lymphomas, nasopharyngeal carcinoma, and gastric cancer. Talking about consequences of the spaceflight, our research during a short-duration space shuttle flight of 10 to 16 days demonstrated that the reactivation of latent herpes virus like Epstein-Barr in 23 astronauts. The radiation of latent infected cells alone is a contributor to the reactivation observed. It is known that spaceflights induces a complicated immune dysfunction in astronauts, and deep space can cause low and medium immune problems. Most of the abnormalities are believed to be strongly related to the microgravity environment in space. Although the interactive effects of radiation and microgravity have been extremely studied since early days of space experiment history, the results vary among experiments. The reactivation of herpes virus is an excellent biomarker to confirm in vivo immune alterations in astronauts, and the spaceflight-associated immune dysregulation may represent a model for patients on Earth that experience secondary immunodeficiencies. Apoptotic cells under microgravity conditions has attracted cancer scientists to look for new cancer therapies, since most cell lines have shown increased apoptosis under microgravity. Also, microgravity provides an environment for cell culture that can induce changes and processes in cells that can be replicated with normal conditions. And this is another reason for the importance of microgravity in different fields such as cancer research, where it can help us understand and suppress the tumor metastasis.
Gamma radiation is included among the types of radiation from the moon that are not present on the ISS to a large extent. A better understanding of irradiation, microgravity, a different Earth atmospheric pressure, large amounts of helium in the lunar environment, and else can influence the reactivation of herpes viruses that should provide important knowledge for spaceflight as well as for terrestrial medicine. The daily radiation dose equivalent on the surface of the moon is around a factor of 2.6 higher than the dose inside the ISS. The reactivation of viruses can occur due to the effect of different types of radiation on latently infected cells. In general, gamma rays are more effective than protons, carbon ions, and iron ions in inducing latent virus reactivation. In space travels, all living organisms are exposed to several unique environmental stress factors. In this experiment, the principal factors are temperature, pressure, pH, and impacts. A slight variation in the temperature can represent the death of the animal cells, so the thermoregulation system of the satellite and the control over it is crucial to maintain these values at the optimal range. Pressure losses will also represent an important risk because of the influence that internal pressure has on oxygen solubility. It is crucial to consider the range of pressure values both inside and outside the system. As for pH, a variation could represent a disruption in the current development of the cells. This experiment presents the mechanized analysis of pH from images taken of the changes in the color of the medium used for cell growth that could indicate variations in acidity or alkalinity. Finally, impacts against external bodies can represent a loss of alternated results. Therefore, it is crucial to have a system of cameras and sensors that monitor the exterior, so that minimal corrections can make in the trajectory to avoid groups of bodies that could represent a threat.
About the concept of operations, as you can see on the table, this refers to the activities to be performed during the 6 weeks of the project life cycle. And then, in the other table, the phases of the mission that appears in sequential order to carry out the project to its end. Now, about the scope of the project, working with the stakeholders and potential clients of the result and data generated once the mission has been carried out, allows us to demonstrate the adaptability of the project and its future usefulness for companies or agencies working in these industries and at international level, as shown in the following figure. About the ground segment, once the launch begins, color images of the cell lines will be transmitted every 4 hours, where possible morphological changes in the cells related with the initial studies done before the launch will be evaluated by the science team, and the data will be received on the Deep Space Network at NASA Jet Propulsion Laboratory, and then analyzed by our project development team, which further represents the link to be performed this mission in the Artemis program.
I'm going to talk about the key performance parameters. First, the mechanism to be used to perform the experiment consists of 6 parts, which are a microcontroller, which will be responsible for making decisions. This microcontroller will be the ESP32D0WD-V3 dual core. The deep cell container, which will be responsible for storing the cells that will be used in the experiment and provide an optimal environment for its study. A microscope, which will be responsible for taking pictures of the cells. The deep cell move system, which is responsible for moving the deep cell container through the microscope to observe and take images of each of the cells. The science control system, which presents a series of sensors that monitors each of the variables that need to be regulated for the optimal study of cells. And finally, the medium change system, which is responsible for changing the culture medium of the cells and extracting the waste generated. Secondly, to carry out the experiment, it's necessary to provide an optimal environment for the cells. So, it's required to maintain the temperature between 33 and 37 Celsius, a concentration of 93 to 96% of oxygen, and a concentration of 4 to 7% of carbon dioxide. To achieve all this, the deep cell container will be used, which is an incubator designed by us. This incubator allows us to control everything mentioned above by using an autonomous system that will monitor each of the variables to be analyzed and keep them within the established range. The system is the one shown in figure, which presents a series of conditions that will allow to entry of different nutrients that the cell will need, as well as the entry of oxygen, carbon dioxide, and the extraction of waste.
The platform required for this mission must meet certain criteria to make sure it is suitable for travel along the cislunar orbit as it is intended. These requirements are the following. For starters, it must support at the bare minimum a 10 kg payload and provide that payload with a power supply of 65 watts or greater. The allocated space for the payload must be 400 by 471 by 155 millimeters, that is to say, at its bare minimum. The bus voltage for the platform must be 5 to 12 volts of direct current, and the platform must keep an inner temperature between the ranges of -20 to 50 degrees Celsius. It must have a bus stabilization system in all of its 3 axes, and the type of communication must be that of deep space missions for its communication with Earth, and AUR type for communication between the bus and the payload. Now, regarding the temperature control subsystem, the payload has a very strict temperature range, which makes this a crucial part of the mission. Therefore, the payload must be enclosed within an isolated system. In the optimal scenario, this would be achieved via an aerogel layer within the inner walls of the platform. And that would give us the best result possible when it comes to thermal isolation. Along this system, the deep cell container has 2 Kapton film heaters attached to its sides. This will make sure that the cell container's temperature is kept between 33 and 37 degrees Celsius via an automated system within the microcontroller.
Due to the fact that the load to be sent in this case is an experiment, it is necessary to have an adequate control of it to keep the cells in an environment that allows taking measurements in an adequate manner. For this purpose, a control system is proposed consisting of a main microcontroller. In the functional architecture, we can see how the science control group is in charge of monitoring the vital variables so that the cells can remain alive and in the necessary conditions to carry out the experiment. In addition, when it notices a deficiency level in any monitored variable, The microcontroller sends a signal to correct it. The medium change control group is in charge of changing the medium when the cells are thawed, and in week 4, the microscope and heat control group is in charge of sending the visual information of the cells so that the microcontroller can store them and eventually send them to the ground. The deep cells move control is in charge of controlling the movement of the deep cell so that the microscope can analyze each of the cells in the specified time interval. Payload power budget. In the payload power budget, we got the table 5 worst case scenario power consumption breakdown of main components, the science control, the move control, the medium change control, and the MCU for a total power consumption of 62.05 watts. In this case, it is not necessary to design a complete circuit since typically the platform provides the necessary bus voltage to power the system. The payload mass budget. In the payload mass budget, we got the table 6 mass budget breakdown in the main components, science control, move control, medium change control, the MCU, and the mechanical structure for a total mass of 9.688 kilograms.
For all of this, we can conclude that the project allows development of innovation where there has never been a project to keep cells alive in space and study them without human intervention, with useful applications on Earth and in space. Finally, it was a really pleasure to share this research in the Mission Idea Contest. Thanks.